announcements. I just want to remind everyone that we're in the middle of this search for a new herbarium director. And this is a tremendous opportunity. So this is a senior level hire to be the director of one of our Berkeley Natural History Museums with obvious ties to the MVZ. And we had one candidate already. We have 4 more to come. The next one is next week on Monday. So the seminars are at non-normal times. They're not in the IB Thursday seminar slot. Each candidate is giving 2 talks. The first one is Monday at 4 p.m. and the second one is Tuesday at 12:30 p.m. And they're in person in 2040 and 2050 VLSB. So I just want to encourage everyone in the MVZ to attend these talks. These are spectacular candidates, and you're going to hear a lot of exciting science. So as part of a community, please make an effort to attend all the talks.
Michael, the candidates give 2 talks each, right? Are they themed?
Are they?
Is there a difference between the Monday and Tuesday talks?
The second talk is aimed at sort of more at the herbarium audience. So the first one is a general research talk, and the second one is aimed more specifically at the herbarium audience and addresses questions related to the collection.
But I would add, it's a general museums talk. Yes, it's focused on herbaria, but it's visions for the museum, visions for collections, visions for use in collections, so relevant to us. Grad students, there's a lunch Monday noon with each candidate. And I'll just say it, vice or co-chair, whatever the hell I am at IB, attendance has been really sparse. We need to up our game to look good to attract these people. So please show up.
Yeah, I would just reiterate that, you know, interviews when you're trying to hire a senior level person are very much a two-way street. So we want to get them excited about Berkeley just as we are excited about them. So Monday at 4, Tuesday at 12:30, there are 4 more talks. The next one is next week. It's Stephen Wright, who is currently the chair of the Ecology and Evolutionary Biology at the University of Toronto. So a major figure in evolutionary biology. Okay, are there any other announcements? Next week, we have another student invited speaker in MVZ lunch, and David, you're hosting, I believe.
Yeah, as part of Virginia's activity, well, actually, she has an announcement. But I sent out a 2 forms. Looks like everyone signed up for 6 p.m. dinner with her next Wednesday, but also there's still some slots available for evenings throughout the day if you guys want to check that out. Santa Cruz. And I would say by Friday, I would like to close up to finalize the schedule.
Okay, anything else? I want to remind everybody, if you ask a question in here, given that we're all wearing masks and there's a Zoom audience as well, speak up so people on Zoom can also hear. All right, I will, Becca.
Yeah, I have Martha's seminar talk here for MVZ. It's called Hidden Dimensions of Adaptive Radiation in Reptiles and Amphibians. She's also giving the IB seminar, and the title for that is Behavior is a Motor and a Brake for Evolution.
Okay, I'm going to turn it over to Jackie, who's introducing today's speaker.
Thanks so much for coming, everybody. It's a really great turnout, the biggest I've seen so far and in person. So it's my pleasure to introduce Dr. Suzanne Alonzo here today. She's our first MVZ student invited speaker after a very long hiatus. So really glad that we could get her in person to join us. Suzanne is actually a UC Berkeley alum. She got her bachelor's degree here in IB, but then got her PhD at the University of California, Santa Barbara. So 2 out of 3 UC so far. Following a postdoc at UC Santa Cruz, she became an assistant professor at Yale before returning to UC Santa Cruz, where she is now a full professor. And Suzanne's research group studies the ecology and evolution of reproductive behavior, including the coevolution of mating and parental care, social interactions and sexual conflict, and animal behavior and conservation. Suzanne's lab group primarily studies fishes, and Suzanne herself has been working on the ocellated wrasse for over 25 years, which we'll hear about later on today. And in addition to her research, Suzanne works on projects related to DEIA and inclusive culture change through various scientific societies. Recently, she helped organize an Inclusivity Workshop at the American Society of Naturalists Virtual Conference. And she was also recently elected as a fellow of the American Association for the Advancement of Science a few weeks ago. So major congratulations on that. And with that, I'll turn it over to you, Suzanne, to give your talk today. Thank you.
Yeah, it's a pleasure to be in here in person. It's the first in-person seminar I've given since the pandemic started. And when I first got the invitation, I thought, oh, I could do this over Zoom, but I would so much rather wait and do it in person. And I'm glad today that I waited as well. It's well been worth it. And thank you for that generous and kind introduction. So what I want to do today is tell you about some work my research group has been doing over the years on the ocellated wrasse, which is my main study system. And what I'm interested in general is sexual selection and the diversity of reproductive systems that we see in nature. And it's fun to talk about sort of the origins of this interest here because they actually started in a class in Berkeley a long time ago. And the thing that fascinates me about sexual selection is it can help us understand both the bizarre and the beautiful in nature. It can help us understand traits that would be otherwise puzzling if we thought about them purely from a natural selection perspective. So if we look at, for example, the beautiful tail of the male peacock, that is clearly not designed to increase survival or your ability to get food or away from predators. And it can really only be understood in the context of female preferences and sexual selection dynamics. But also things like sexual cannibalism and the redback spider, group work done by Mady and Andre and colleagues, has shown that it's actually beneficial for the male to be consumed by the female after mating. And so sexual selection has this power to explain things in nature that would otherwise be quite difficult to understand. And the particular thing that I'm very interested in is how interactions within and between groups of individuals shape evolutionary processes and lead to patterns that we can understand in nature. So for example, we've known for a long time that male-male interactions, male-male competition drives the evolution of reproductive traits. And similarly, male and female interactions, male-female interactions drive the evolution of things like preferences and courtship displays. But what I've worked on for quite a while now is the fact that you actually have to think about all of those interactions simultaneously in order to understand the reproductive traits that we see in nature. So if you look at only one of these interactions in isolation, you actually miss a lot of the important evolutionary dynamics and have an incomplete understanding of what you see. In addition, in my lab, we're really interested in the interplay between evolution and plasticity. So we're interested in evolutionary change over long time scales and also very rapid behavioral responses to these interactions and how those two in combination drive patterns of selection and help us understand what we see in nature. So this is the conceptual framework that I carry around in my head. And so if I look a little distracted, it might be because of the complexity of things that we're trying to incorporate here. So what I want to do today is first introduce you to the ocellated wrasse, the species that I've been studying for over 25 years now, and talk about two different studies. The first one focuses on the neuroendocrine mechanisms underlying the behaviors that we've been studying and how they relate to mating success. And then the second study I want to tell you about looks at how social interactions can influence patterns of sperm competition and sperm allocation and sperm trait evolution. And then I'll finish with just some brief general conclusions. So what you're looking at here is a photo taken from my field site in Corsica. Sadly, I've not been there in almost 3 years thanks to the pandemic, but it's a beautiful place to go. And if you were to join me on scuba and jump in the water here, what you would see is an incredible diversity of fishes. And in particular, there are 9 species all in the same genus, the Symphodus genus, that coexist in that location. They have the same basic ecological niche. It was a rapid radiation after their common ancestor came into the Mediterranean. And although they're very similar ecologically, they have incredibly variable social systems and mating systems. So some are very dimorphic, some are not dimorphic at all, some have parental pair and others do not. And so there's lots of diversity in this system. And I've been studying this group, and in particular, the star here in the middle, the ocellated wrasse, for quite a long time. And so there's not just variation among these species, there's also incredible variation within the species, within the ocellated wrasse. And this is what first drew me to them actually as a graduate student a long time ago. There are 3 alternative male types that coexist in this species, 3 different ways of reproducing as a male. We have nesting males, bright, colorful, they defend territories, they build nests out of algae and court females who come to those nests to spawn. We also have sneaker males, which are the classic parasitic spawning strategy. They don't court females, they don't build nests. Basically, all they do is hang around nests and try to sneak spawn. They rush in when the female and nesting male are spawning and release sperm. And then perhaps most fascinatingly, we have this intermediate male, a satellite male. They are intermediate in both size and color. They don't build nests, they don't defend their own territories. They actually associate with a nest and a nesting male, but only for a short period of time. It's a short-term association of a matter of days. And they're both a cooperative and a competitive strategy. So they cooperate with the nesting male by chasing away sneaker males, but then when the nesting male is mating, they also sneak spawn. So they're both a reproductive competitor and a cooperator in terms of their interactions with the nesting male. The other thing we know about this system is that females show a strong preference for nesting males. So they prefer nesting males over sneaker and satellite males. And the paternity, the share of paternity among these males sort of maps onto that preference. Nesting males sire about 2/3 of the offspring in a nest on average. Satellite males get the next closest share, which is about 10 to 15%. And the rest is divided up by this among the sneaker males. And as many as 20 sneaker males can surround a single nest where there's really high mating success. So there's a real skew in paternity. And the risk and intensity of sperm competition that these males experiences also depends on their mating role. So nesting males sometimes are pair spawning, spawning singly with a female, so not in competition with other males, and are sometimes joined by other males. Satellite males are usually joining the nesting male and the female. And sneakers are often one of many sneaker males that are releasing sperm. So the risk and intensity of sperm competition increases as we go from nesting males to satellites to sneaker males. They have external fertilization. The nesting males provide parental care. So that's an important component of the system. And they breed seasonally in the Mediterranean in roughly May and June. So what I was first interested in was the maintenance of these alternative male types. And one of the things that if we look at these nesting males, I don't know about you, but to me, they're bright and beautiful and really striking. And we know that females prefer them over the nesting male types. So we might also think that there's strong sexual selection among nesting males. And so that was one of the first things I was interested in, is are females preferring one type of nesting male over another? And what you're looking at here are the frequency distribution of spawning or mating. And this is per 10-minute observations from a bunch of nests chosen at random, just sort of natural spawning patterns. The blue line is what you would expect under random mating based on the assumption about fossil process. And the orange-ish red bars are what we actually observed. So this is one of the first things when you study sexual selection that you have to ask, is there any evidence of a skewed distribution of mating success? And what you can hopefully see from this graph is that there's evidence of a very non-random process going on here. Some males have incredibly high mating success. Many males have very low mating success. So it looks like a classically sexually selected species. So the natural follow-up is you start to ask, is it this color pattern? Is it this behavior? Is it something about the nest? Is it something about the territory? And what I can tell you after a lot of hard work and time underwater is that actually the females are not paying attention to any of the male characteristics that you might expect. None of the sort of nesting male specific characteristics explain any of the variation in mating success. So they strongly prefer the nesting male phenotype, but once it gets to choice among nesting males, there's no evidence of selection on those particular traits. And in fact, if you track individual male identity, the male identity is not a significantly explanatory variable. And so something else is going on in this system. And so what we've become really interested in over time is actually how the social interactions and the social environment explains this skewed distribution of mating success among nests. The other really interesting thing about this system that I just want to underscore, because it's sort of motivated the two studies that I want to tell you about, is that the fact that you have cooperation between the nesting male and the satellite male, it's a mutualism, they're unrelated to one another. This is not an altruistic action. This is a mutual benefit situation where they both benefit from the interaction. But these are unrelated individuals who do not have a long-term bond of any kind or long-term interaction of any kind. And so there's this interesting interplay between cooperative interactions or positive interactions and competitive interactions simultaneously in that system. And so it's interesting to think about how that modifies our expectations about patterns of sexual selection, patterns of aggression, submission, things like sperm competition. So I just want to underscore that we're interested in how this interplay between cooperation and competition might affect sexual selection. Okay. So before I tell you about these two studies, I'm going to try to summarize a lot of research in a short period of time on this system. It is a relatively complex mating system. That's what I find very fascinating about it. But I'm going to do this with a little bit of a sort of cartoon, but I want you to just hopefully trust that there's a lot of science, a lot of data to back up what I'm telling you here today. So imagine, if you will, that there is an ocellated wrasse female shown here in red, choosing among nests. So I already told you she's not choosing among those that nest based on the characteristics of nest or the characteristics of the male. In fact, what she pays attention to is she pays attention to the presence of other females. So we have strong evidence of female mate choice copying going on in this system. So females go where other females have been before. And as a result, when we think about sneakers and satellites mapping onto that distribution of females or choosing where they're going to attempt to mate, the satellites and nest females also map onto where the females are, right? They're going to go where the mating success is available. And so when satellites and sneakers are choosing among nests, they also tend to choose where there's higher mating success and higher mating opportunities. And so these social interactions that start with female mate choice copying basically drive that skewed distribution of mating success. So it's actually the social interactions themselves that lead to that skew that I showed you before. Once you have this skewed distribution of mating success, you end up with nests that have many females, but also many sneakers and satellites. And so there's very complex social interactions that are going. And what we know is that this likely led to the system of the satellite males because the satellite males play an important role in reducing sperm competition for the nesting males. So there's a lot of social interactions that are underlying the sexual selection dynamics in this system. And so one of the things we've become really interested in is not sexual selection on the color pattern or the displays or the classic traits that you might study, but what is sexual selection on social plasticity, the ability to respond to variation in the social environment. But if you're a female choosing among the nest, you're looking not at the male traits, not at these fixed male traits, but instead at the social environment at these other nests. If you're a sneaker male, figuring out when and where to spawn, it's a response to the social conditions, not a response to sort of fixed traits or characteristics of your competitors. And I think one of the best ways to get across the importance of these rapid behavioral changes is by actually looking at a quick video. Hopefully this will work. So what you're looking at here is a nest and the nesting male in the center. You just saw a spawning event. You can hopefully see that I think 3 or 4 males were involved there. The satellite male is so the nesting male is this bright colorful guy. The next biggest male that you see them interacting, being both aggressive and submissive, and spawning is about to happen, I believe. And so the satellite male and nesting male, the satellite male just snuck in. So he got away with the mating success. So I show this just to get to the point of, when we're thinking about how sexual selection is shaping the traits in this species, we're really thinking about these rapid behavioral responses in response to relatively subtle interactions and opportunities as they arise.
Did the sneaker males look like the females?
So the females are small and drab like the sneakers, so they look superficially similar, but they have a genital papillae, which is in a sort of shape to their body, which is significantly different. So you can kind of pretty easily spot them underwater. The females also, the genital papillae is darkened. And what I can tell you is they can definitely tell each other apart. So it's not a true female mimic. That's a great question. Yeah, so superficially they look like a female, but behaviorally they respond to one another quite differently. Yeah. So one of the things that we've been interested in is this sort of sexual selection on behavioral plasticity or social plasticity. And usually when we think about phenotypic plasticity, we think about that being some phenotype that's responding to natural variation in the environment. But the thing I really want to underscore here is that we're really interested in this case, I'm really interested in how an individual's behavior is changing over time in response to the social environment or social cues. And so one of the first really questions that we wanted to ask is, what are some of the mechanisms underlying their capacity to produce these behaviors? So the first study I want to tell you about actually is focused on understanding some of the neuroendocrine mechanisms that are underlying the social behaviors. And one of the reasons for doing this is we wanted to do basically phenotypic engineering. We were interested in altering the phenotypes using these mechanisms. So the focus was, the mechanisms are super fascinating. I love learning about them, but they're really an avenue for doing manipulations, experimental manipulations, where we can alter the interactions, behaviors that we see in nature. And so we've done a wide variety of things looking at the differences between the alternative male types. But the study I want to tell you about today actually focuses on the nesting males in particular, and understanding the mechanisms that may be underlying some of the behaviors that they exhibit. So as a bit of background, when we compared those alternative male types, we found, for example, that there are significant differences. This is no surprise, but just to give you the background that we kind of know what we're doing. Nesting males have higher levels of 11-keto-testosterone, which is the fish homologue of mammalian testosterone. And so as you would expect, the more dominant and aggressive males have higher levels of circulating testosterone. Similarly, if we look in 2 of the key brain areas that are associated with vertebrate social decision-making, we see significant differences among the male types in the gene expression of the androgen receptors. This is the preoptic area and the dorsolateral telcephalon. So satellites and nesting males have significantly higher levels of this androgen receptor signaling. So what this told us is that androgens, no surprise, are very important in male behavior and male interactions. And so we decided to do a manipulation under natural conditions in the wild on the circulating levels of androgen. So what we did is, and this work was led by a postdoc of mine, Bridget Nugent, back when I was at Yale. And so what we did is we go out and observe nesting males at their nests, do a detailed observation of the nest. We then catch those individuals, inject them with flutamide, which is an androgen receptor signaling blocker. We'd mark them and we'd release them back in the wild. And we would also have a sham manipulation. So we have a vehicle and a true manipulation. Then we would observe them over the next few days on a daily basis, doing detailed observations of the nest again. Then finally, we would collect them, we catch them, which is sometimes the hardest part because they're easy to catch the first time, but hard to catch the second time. And then collect a variety of tissues as well as the nest and larvae to count the number of larvae and do paternity analysis. And this is our beautiful field lab where we did some of these brain and blood collections. And this was done in collaboration also with my long-term colleague, Kelly Steiber and Hans Hoffman at UT Austin. Okay, so the first 2 sets of slides, the next pieces of data I want to show you are simply to show you that the manipulation worked, right? We're doing this manipulation in a non-model organism. We're doing it under natural conditions in the wild. We really want to make sure that this injection that we gave them actually had an effect on their physiology. So there's going to be a lot of slides where the flutamide, the true treatment, experimental treatment, is shown in blue. The vehicle is shown in white. And we had a significant effect, as expected, on the circulating 11-keto-testosterone. There's also, in a 3-day period, a significant effect on the relative gonad size. So males that were treated with the androgen blockers, gonads were smaller, less weight. And there was also a significant effect on sperm velocity. So we're having a physiological effect. We also had an effect on patterns of expression in the brain. So in particular, this is the androgen receptor that I already told you about. If we look again, in this case, in the preoptic area, there was a significant effect of gene expression in the preoptic area. We also found a significant change in the vasotocin receptor expression, which is an important vertebrate social signaling pathway. So we affect the bodies and the brains. What happens to the behavior? Which is the thing I'm really most interested in, to be honest. Okay, so the first thing that we found, perhaps a little bit surprising, is that if we look at total aggression, the number of times that nesting males chased conspecifics in the observations following the manipulation, so we're looking at a change in behavior before, after, because there's lots of natural variation. And all of these behaviors were collected in underwater in the wild, just to underscore that. We found no significant difference. So manipulating this, blocking the androgen receptor signaling pathway did not have a significant effect on total aggression. Somewhat surprising, because you might think that it would decrease aggression because you're blocking the androgens. But when we looked at who the aggression is directed at, whether or not it was directed more towards sneakers or more towards satellite males, for example, we did find a significant difference between the 2 treatments. So this axis that you're looking at is a change in the direction of nesting male aggressive behavior, which is a little bit tricky to think about. This is basically, was there an increase in the aggression towards sneaker males, or was it an increase in aggression towards the satellite males? Did they shift to whom they were directing their aggressions? And in the sham manipulation, there's actually no significant change in terms of whether or not they shift their aggression. But this flutamide, the males that were treated with flutamide that had their androgen receptor signaling pathway suppressed, actually showed an increase or a redirection, if you will, of their aggression towards the satellite male. Now, I'll remind you that the sneaker males are the ones that are coming in and are not a cooperative partner, whereas the satellite male is actually in a cooperative interaction, also competitive interaction with the nesting male. And so this redirection of the aggression has the potential to have a significant effect on the mating success of the nesting male, but it also has a potential to have a significant effect on the social dynamics of the nest. And that was one of the things that we were interested in as well. Another effect that we saw sort of downstream from that manipulation was that there was a change in sneaker male behavior. Now, the sneaker males were not manipulated in any way, right? They were not touched. We didn't do anything to them. But of course, they were interacting with a partner that either had their androgens suppressed or did not have their androgens suppressed. And what we saw is that this is again a before-after comparison. So the change in sneaker male submissive behavior was not significantly different from zero when you talk about the control or the vehicle. No change in behavior there. But the sneakers that were interacting with a male who'd been treated with flutamide actually were much less submissive to that nesting male. So there was a shift in sneaker male behavior as a result of this manipulation. And if we look at the effect of that sneaker male, that change in sneaker male submissiveness and the spawning rate at the nest, this is a difference, again, a before-after comparison. There's actually a significant correlation between that change in sneaker behavior and the change in spawning. So less change, higher spawning, more change, meaning sneakers were less submissive, a greater decrease in the spawning. So less mating success when sneaker males are less submissive. Now, to be completely clear, we did not find a main effect of treatment. This is a correlation among the phenotypic variation that we generated. So there is some noisiness in the data, but there's also a lot of variation in the sneaker males themselves. So I think that this is an interesting way of thinking about. If you think about phenotypic engineering, it's not just about treatment effects, it's about generating phenotypic variation. And when we look at it as phenotypic variation as a whole, we find a significant effect of this change in the social dynamics of the nest on mating rate. Similarly, if we look at variation in what's going on in the brain, we find a significant relationship between that androgen receptor signaling and the change in spawning rate. So again, what's going on in the brain, the manipulation had an effect on brains, and that also had an effect on spawning success of these males. Okay, so we were able to manipulate the neuroendocrine mechanisms in these nesting males and see consequences not just for their behavior, but also for the behavior of their social partners, with consequences for the spawning success of all individuals involved. And so what I would like to posit is that we were able to document variation mating success, which is how we define sexual selection, on those neural processes underlying social interactions. So we generated variation, and we saw that variation has consequences not just for their behavior, but also for the mating success of the individuals involved. And so the follow-up studies that we'd like to do are looking at this more under natural variation in the wild. So hopefully in a few years, I'll be able to tell you more about that. Okay, so that was the first study I wanted to tell you about. The second study I want to tell you about focuses in on sperm competition in this system. So as I mentioned, the risk and intensity of sperm competition that these males experience changes across those alternative, or differs across those alternative male types. And so we've done a variety of things to look at sperm competition over the years. And one of the things that I think is really important about this system for studying these questions is not only do we have the classic alternative male types that are relatively well studied, but we have this interesting satellite male who is not just a competitor, he's also potentially a cooperative partner in the system. So just to give you a little bit of background, some things that we know about sperm competition in this species. So this is the graph I'm showing you here is the sperm number, the number of sperm that were released in a single spawning event when there was either a nesting male plus a satellite male, or a nesting male plus a single sneaker male. And mainly what I want you to see here is that although nesting males alone release about 1 million sperm, nesting male and a satellite together release about 2 million sperm, but adding a sneaker brings you up to about 5 million sperm. For reasons that I don't need to go into now, we're pretty sure the nesting male doesn't know. He can't change his sperm release in response to who's joining him because he spawns first. And so you can think about this as being evidence that the sneaker male release 4 to 5 million more sperm than do either a sneaker or satellite male. So sneaker males are at an advantage in terms of the number of sperm that they're releasing in a single spawning event. Some of the other things that we've learned over the years are that when sperm competition is high, which is cued by the presence of a large number of sneakers at the nest, nesting males don't release more sperm, so they don't increase the number of sperm they release. They release even when there's lots of competitors present. But what they do is increase their aggressive behaviors towards the sneaker and satellite males. So they invest in behavioral changes rather than changes in sperm release. And another really interesting strategy that the nesting males adopt is they become temporarily unwilling to spawn with willing females. So they actually shut down spawning at the nest. Just another piece of background before I tell you about this one particular study is that we've also looked at how interactions between the sexes affect patterns of fertilization under sperm competition. So we know that higher velocity sperm are favored. They lead to higher patterns of paternity. And also females not only exhibit copying, like I told you before, they exhibit cryptic female choice. So the ovarian fluid of the female affects a variety of sperm characteristics, but it also decreases the importance of sperm number, which in essence takes away the advantage that the sneaker males otherwise would have. And so it sort of indirectly leads to higher nesting male paternity. Okay, so the particular question that I wanted to tell you about today is that we were interested in whether or not the alternative male types differ in both sperm traits and ejaculate production or sperm allocation. And I'm going to focus on sperm allocation today, although we also found some interesting results with respect to the sperm traits that I'd be happy to talk about. And so the interesting thing here is that if you think about these alternative male types, there's a variety of different predictions you could make if you're comparing the 3 different types. So the first way you might think about this is that those sneakers are experiencing higher sperm competition. And so what classic theory would predict then is that they should allocate more, they should be producing more sperm, and if possible, faster, better sperm. That's one prediction you could make based on the literature. However, another prediction you could make is that we also know that nesting males are larger and older and in better body condition. That's just some information we have about the species, which means you might actually predict that they should produce more and higher quality sperm because they're higher quality individuals. And then if you take into account the satellite males who are somewhat cooperative and somewhat competitive, I would argue we don't have the theory yet to predict how that might modify the patterns you'd expect to see in nature. What we know from cooperative breeding systems is that the subordinate males who are cooperative often have reduced, if not completely shut down gonads, so they produce much less sperm. But of course, if you think of the satellite male as like the dominant sneaker male, he might actually produce a higher or more sperm than a sneaker male. Okay, so what we did is we went out and we caught lots and lots of different males of known type. Across a variety of years, we took the gonads and we weighed their gonads and we weighed their bodies. And we wanted to look at the allometric relationship, right, between body size and gonad size. And so what you're looking at are the results of that data collection, and the lines are the best fit model that we use. This is a, oh, forgetting, I have to look at the paper. It is not an ANCOVA because the data did not conform to the assumptions of the ANCOVA. I actually forget which it is. And so if we're looking at the relationship between the natural log of body weight or soma weight and gonad weight, what I can hope you can see is that the 3 different males have a different relationship. This is that allometric relationship between body size and gonad size. So all else equal, the nesting males have lower investment relative to their body size in the gonad, the testes. And the satellites actually have the largest absolute testes. So if I were to show you just simply testes weight, what you would see is that the satellite males would actually pop out with having the largest testes not largest relative to their body, largest total testes compared to either sweeper or nesting males. And they also have a significantly different allometric relationship between body size and gonad size. So I think one of the clear things we can see is nesting males do have a lesser relative allocation to their testes, but these reproductive cooperators are not that cooperative when it comes to their sperm production. So the other thing that we were interested in was sperm regeneration. If you think about the idea that maybe the nesting males are a higher condition, higher quality male, you might imagine that they can actually regenerate their sperm at a faster rate. So what we did is we plot live individuals. We took a sperm sample from them. We looked at sperm volume, cell counts. Sperm volume, cell counts, sorry. I was thinking there might be a third thing. And then we waited 5 hours. We kept them in tanks, waited 5 hours, and then we took a second sample from them. And we were curious whether or not there was differences in total production. So the data I showed you earlier was about individual spawning events. This is the total production of sperm that they have at any one point in time. And we were also curious if they might differ in the rate at which they could regenerate a new ejaculate. And so this graph shows you the total volume of the sample, which is both seminal fluid and sperm cells, the sperm concentration, which is the number of cells per volume, and then our estimates of total sperm number from those two variables up above. You're looking at the initial sample and second sample for all 3 male types. So the main thing you can hopefully see is that if we compare the initial to the second sample, perhaps no surprise, there's a reduction in the sample. This was 5 hours apart, so basically 5 hours is insufficient for them to regenerate a new ejaculate. So that's interesting given the fact that they are releasing, millions of sperm. Like some of the males are mating 60 times in a 10-minute period and releasing somewhere between 1 and 5 million sperm per time. So there's a huge investment in the sperm release and sperm production. Also, sneaker males produce a smaller ejaculate than do the other alternative male types, which again is interesting given that they're releasing more sperm per individual mating, but they have fewer mating events in total. But there was no significant difference between the alternative male types in their rate of regeneration. So satellite males have smaller ejaculates. They're all having a hard time reproducing them or regenerating them, but there's no significant difference among the male types in that capacity to regenerate. All right, so we found that there are significant differences in sperm allocation and ejaculate traits. I didn't have time to tell you a lot of the sperm traits, but there are also sperm trait differences between the male types. But what I think is interesting to think about is that what we find is not always what we would predict based on current theory. So a lot of the theory that predicts how males should allocate among sperm production or in response to sperm competition is based on pretty simplistic assumptions about what those males are like. It thinks about discrete, nesting male or dominant and subordinate males. It doesn't include any of these sort of complex social interactions or social roles. And so that's one of the things my lab has been thinking about is can we actually develop some theory that's a bit more nuanced and thinking about how males might allocate. The other thing that I would argue is that the social roles that these males inhabit in their alternative male reproductive tactics influences their sperm allocation and sperm traits in interesting ways. And so the social interactions themselves are likely selecting on those reproductive traits, and perhaps there's also a feedback between male-female interactions as well as a result of cryptic female choice being important in the system. So those are the 2 main studies I wanted to tell you about today. But one of the things I've been thinking about a lot, perhaps because I haven't been able to go in the field, is, what are some of the things I've learned over 25 years from the ocellated wrasse about sexual selection. And so one of the things that we've learned over the last 25 years is that there's a really complex and important synergy between intra-sexual and intersexual selection. So I would argue that when we think about any particular trait, we need to consider interactions both within and between the sexes simultaneously. The other things that we've learned over the years is that social interactions can drive patterns of sexual selection. And so when we think about the classic theory that looks at a female preference and a male display trait, it really doesn't capture how the social interactions are playing out in that system. And I think we might get some different predictions if we start to think about the nuances of how that kind of decision-making and social interaction plays out in real systems. The other thing that I've spent a lot of time thinking about is the role that reproductive cooperation and competition play in systems. There's always elements of cooperation when reproduction is happening, right? Both individuals need one another in order for reproduction to occur. But competition among individuals is also a really important factor. And the balance between these sort of positive and competitive interactions and systems likely determine which traits are favored by selection. And I would argue that our current theory really doesn't incorporate that kind of thinking in a very nuanced or deeper way. And the other thing that I've become really interested in is that sexual selection is shaping so many aspects of the organism, right? When we think about sexual selection as just explaining things like the peacock's tail or some, competitive, some defense trait that an individual has that's involved in intersexual selection, we're really missing the ways in which sexual selection shapes a lot of the biodiversity that we see in nature. And so we've been thinking a lot about how sexual selection affects things like patterns of growth, which is one of the drivers of the alternative male types, gene expression in the brain, physiology, things like personality, and also whether or not and to the degree to which organisms are plastic is also likely shaped by sexual selection. And so, my takeaway, if I leave you with one thing that I'd love to have you think about more, is that sexual selection can really explain much more than the bizarre and the beautiful. It's actually one of the sort of drivers of biodiversity. And I think it's influencing what we see in nature in a much more nuanced and powerful way than we've yet to appreciate. So I think there's a lot more we can learn. We know a lot already, but there's a lot more that we can learn in the years ahead. So with that, I just want to thank my many wrasse collaborators. The first project that I told you about was led by Bridget Nugent, a postdoc in my lab. And the second project I told you about was done in collaboration with Kelly Stiver and Susan Marsholo, long-term collaborators on the wrasse project. Lots of people have helped in the field. My funding sources are always appreciated. And all of you for coming out to an in-person seminar. Thank you so much.
Great, well, we have time for questions, so I'll open the floor, maybe first in person, and then we'll check the Zoom chat. I'll let you just call on people.
Thanks so much for that really interesting talk. It's so neat to see about these complex social interactions in fish. I was wondering about female behavior and how that might be modified by her perception of who's had success with the spawning event, if it's the nesting males or if she's perceived kind of a lot of activities from sleeper males. So I know you showed there's like ovarian fluid, and I guess that's maybe she's perceiving the males that are around her and then changing her investment in the spawning event. If I understood that correctly, but correct me if I'm wrong. But then after the spawning event, I don't know if there's any parental care in this species and if she maybe modifies that, or subsequent engagement in spawning events or number of spawning events. But what do we know about the female?
Yeah, lots of great questions there. So I'm going to repeat the question for those in Zoom. The question was about what we know about female responses to in terms of both mating and cryptic female choice after mating or during mating. So we know, what I know is that the females cue off other females. So one of the most significant explanatory variables that we find, no matter what kind of study or analysis we're doing, that explains mating success is recent mating success. So if a male has had a lot of mating success in the last hour or the last minute or the last half a day, it's a significant explanatory variable of future mating success. And also, independent of that, if one female is present at the nest, it has a really significant large effect on the probability that another female will spawn there. So they're likely cueing off the eggs, although we don't know exactly what the cues are there. Olfactory would be my guess, but they might also use visual cues. But they also literally cue off the behavior of other females, so they're watching one another. So that's the mate choice scale. When it comes to the ovarian fluid, we're actually starting to look at plasticity in ovarian fluid, whether or not they adjust the ovarian fluid. What we know so far is that the ovarian fluid in general reduces the importance of the number of sperm, which then indirectly favors nesting males. So it's not that they're favoring nesting male sperm, it's that they in essence, the ovarian fluid reduces the advantage the sneaker males would otherwise have in fertilization. We don't know if they plastically change their ovarian fluid in response to the conditions at the nest. The ovarian fluid studies were done in the lab. You get like microliters of ovarian fluid. You know, like in salmon, you can get like buckets of ovarian fluid, and in wrasses, you get microliters of ovarian fluid. So it's, so far, they've all been lab studies. We also know the ovarian fluid influences things like motility, velocity, and chemotaxis. And in fish, there's a micropyle on the egg. So there's a single entry point to the egg. And so ovarian fluid basically also operates as a chemical gradient that male sperm use to find the micropyle, which is what they need for fertilization. So we're following up with some studies also about whether or not the nesting male, the different types differ in their capacity to exhibit chemotaxis, or if the ovarian fluid somehow influences their ability to exhibit chemotaxis. Females do not provide parental care in the system. So I should have said that. It's male only parental care. So basically females come in, leave their eggs and go. And so there's not a lot of potential for them to influence development of the offspring or parental care after. But they do, by copying one another, they increase their probability of getting parental care, because the most significant factor influencing whether or not a nesting male provides parental care or not is how successful he's been. And so we've actually done some experiments that show if you bump up his mating success experimentally, the probability that he'll stay and take care of the offspring goes up significantly as well. So there's a clear direct benefit to the female of choosing a successful male.
Thank you.
Yeah. I think you're in.
I had a question about the development of the different male types. So is it something that's like plastic and determined by environmental conditions, or is there like a predetermined genetic component to it?
Yeah, great question. So it's size dependent, but they can switch between types. So in this species, looking at otoliths and fish, you can age them. They're either 1 or 2 years old. And so individuals that have slow juvenile growth, so pre-reproductive growth that's slow, tend to be a sneaker in their first year and a satellite male in their second year. Individuals that have rapid or higher pre-reproductive juvenile stage growth will start as a satellite male in their first year and then become a nesting male in their second year. So nesting males are all 2-year-old males, sneaker males are all 1-year-old, and satellites are mixed. So there's sort of 2 life history pathways, even though there's 3 types, because the ocellated wrasse never does it simple. And so we don't have any evidence that it's genetic polymorphism. I can't completely rule out the fact that there might be genetic variation driving differences in growth that then drive the life history pathways, but we know that size or growth, one or the other, or some combination thereof, is what leads to the separation of the different types. Yeah, and they can change over their lifetime, so it's clearly not a simple genetic polymorphism. Yeah.
So if I understood the layout, the framework, it's so complicated. It's a great talk, by the way. It seems like you assume that there's some randomness to this process. Like if a male happens to be lucky enough to attract females, that it becomes this positive feedback loop. And I'm just curious if you've had a chance to actually see this in action, where a male is successful this day or this week, and a different week, a male that's not having any matings is now the one that's successful. So that you can really see that there isn't some trait that the male has that the females are queuing into. Is this, can you actually see that, or season to season?
So I'm going to try to summarize your question. I think I understand it, but I'm going to try to summarize it, which is for the Zoom folks, which is that, do we understand sort of what sets off this positive feedback loop initially? So what
does it come and go?
So what I can tell you is all of the data that we have is consistent with random choice at the first mating. And we're able to track males over multiple spawning cycles, and male identity never explains any of the variation across cycles. It explains nesting males' recent mating success explains their next moment of mating success, but they go through a spawning cycle where they build a nest, they spawn in it, and then they provide parental care. And once you move to a new nest, all bets are off. And so the same male can be incredibly successful on one day and not on the next, but he could also be incredibly successful in one cycle and a complete loser in the next one in terms of no mating success. And the mating success is really skewed. So there's a lot of males that get no mating success, desert their nest because there's nothing to take care of anyway, and then go on and try again. I once tried to get a paper published with the title "Good Luck, Not Good Genes," but they didn't let me keep it. but it's consistent with complete randomness at the beginning. Yeah. I'm sorry, I lost track. Go ahead.
So I'm kind of following up on Jim, but at the other end of the process, and that is, I was wondering if there's a point at which, since males are providing care, their nests are full. I'm thinking kind of like a two-spotted goby thing where there's that temporal pattern that, you know, eventually that changes what females are choosing males based on. It changes the dynamic of sexual selection. Those kinds of things.
Yeah, it's a great question. So the question is whether or not, in essence, the nests become too full then, and then whether or not the male's parental care becomes limited. So in other fishes, we've certainly found density dependence in parental care where you can get too many eggs. In the wrasses, I've never found any evidence of it. So if you collect their nests, you know, there's like super high fertilization rates, super high development rates, even when there's, you know, and the range can be literally 0 to like 50,000. You know, it's like many, many, many eggs. So they seem to be capable of taking care of a very large number of offspring. And we've never found any significant differences in like hatch rates or anything like that with density of eggs. The main thing that the males are doing in terms of parental care is chasing away predators, both conspecific and other species. There's also invertebrates that prey on the eggs. That's like thing number one. And then oxygenation. So a really common thing in fish is that they fan the eggs to oxygenate them. You might expect that that would lead to density dependence, but we've never found any evidence of it. The interesting thing that I don't have an explanation for that they do is they go through these, if it's purely a positive feedback loop, once you're successful, you should just stay put and keep on attracting females and keep on letting the eggs develop if there's no upper limit. But after 3 to 5 days of spawning, depending on the water temperature, they shut down and the nesting male refuses to spawn anymore and then becomes parental. They shift in a completely different phase of behavior. And why they do that is actually an open puzzle for me. You know, maybe the nest starts to degrade. I don't know, but there's a pretty clear switch in their behavior, and they, you know, females will still be coming to the nest and poking around and trying to spawn in there, and the male literally body blocks the nest. So why they do that, I'd love to hear hypotheses. Yeah, go ahead. Yeah, go ahead.
I'm just curious if the first, you know, mating is somewhat random, whether there's spatial arrangement complexity among different males in an area where you could imagine, you know, different orientations favoring that initial event.
Yeah.
In a spatial context.
Yeah. So the question is whether there's not a sort of spatial pattern to mating success, and you might expect clumping. So what we find is that there absolutely is spatial clumping of mating success, but not in terms of a fixed location. Instead, what we see is if we map the nest and we look at patterns of mating success, there's a transfer of mating success. So as and we've done experiments to get at this too, you can actually block a nest and then see where females go. If you take a nest out of circulation, if you cover it up, which is what happens naturally when the male becomes parental, when he shuts down his nest, is that females they choose among the surrounding nests. And so you get this local transfer of mating success where you kind of get hot spots. But they're not fixed hotspots. They're basically moving throughout the spawning season. And we have XYZ coordinates on all the nests for the last decade or so. And that the XYZ coordinates themselves, the fixed location, never explains any of the variation of mating success either. Yeah, I think Chris is
sure. Yeah, no.
I'm trying to track, but I'm doing badly.
Yeah, no, fantastic talk, fantastic answers to all these questions. We all have asking questions all day long. Yeah, kind of building on Michael's earlier question, but I mean, what maintains the stability of the satellite males? Why haven't female mimics? Because there is so much variation in life history strategies. Yeah. It seems like there's always seeker males, but then this third strategy, why do we see it here?
Yeah, so the most honest answer is I don't know. What I can tell you about the whole system is that in the genus, there are some species that have no alternative male types. There are a few that have 2 types, so the typical dominant subordinate male types, and there's 3 that have something like the satellite male. So I really want to study the whole system to look at sort of if we can think about what are the common conditions when you get the satellite male types. What I imagine in my mind is that there's this strong, well, so what we know is there's a strong preference for the nesting male. So if I were to time travel, if I could time travel and watch the evolutionary process, what I hypothesize might happen is that you initially had strong female preferences on continuous male variation. And that sort of led to this nesting male phenotype, like it's almost a step function, right? If you're a nesting male phenotype, you're good. If you're not, the females do not like you. And so there probably was continuous variation early on, and that strong female preference for that nesting male phenotype, which is a clear limit to their mating success. They need, if it's obligate parental care. So if the nesting male deserts, those eggs do not develop. That strong female preference led to the origin of the sneaker male, like no surprise there, as what males did when they couldn't produce the nesting male phenotype. Best of a bad situation, not a very good term, but that kind of concept, given that they couldn't produce the nesting male phenotype. And then my guess would be that over evolutionary time, we would see the satellite arise as the third type after those two are in existence, because there's an advantage to the nesting male, because we know experimentally that he decreases the risk of sperm competition that the nesting male experiences. And then also the satellite male is tolerated by the nesting male. He's allowed closer to the nest. So he has higher mating success and also higher paternity. And probably only the largest sneakers could adopt that strategy. That's my hypothesis. How to test it? Time travel.
How many times does a male mate in its lifetime? And the females visit multiple nests?
Yeah.
And,
So-
and the last question related is, can you track an individual for its lifetime?
Yeah, so we track almost everything you saw was on marked individuals that we track throughout their lifetime. So we give them a elastomer tattoo. Survival rates are low, so tracking them their whole life usually means at most 2 years. But most of them, it means 1 season. So you asked a couple of questions about
How many times does a male mate?
how many times does a male mate? So there's incredible variation. Males go through somewhere between 3 and 5 nest cycles, which are on average about 7 to 10 days. They can mate between 0 and 60 times per 10-minute period. So there's huge variation. So if a male was super successful, he might mate at that high rate for 2 or 3 days in one of his cycles. So they can mate, thousands and thousands of
And
times.
those cycles, are they within a season?
They're within a single season. So nesting males you're only a nesting male for a single season. So they're all 2-year-old males, and we have never found a 3-year-old male. And females usually release about 30 to 50 eggs per spawning event, and then might spawn, we might see upwards of 5 to 10 times on a given day, but they mate every other day or every third day. And they certainly move between multiple nests. There's high levels of multiple mating in terms of both, not just paternity at a nest, but moving across nests.
I was kind of wondering about the allometric scaling of these hormones and body size, because you might imagine I'm imagining a situation in which you see this sort of variation in life history with increasing age, where the body size of the fish increases with age. They then in turn have more of these hormones which modulate the behavior, which manifest as these.
So you're talking about like circulating androgens or circulating hormones of any kind. Yeah, so we have size data on all of the males that we've taken hormone data on. I can't remember off the top of my head if there was any relationship with the size. I think the one hormonal thing that I remember where size came out significantly as an explanatory variable was actually that, oh gosh, I'm good. I don't think the satellite males vary in size. And I think that that the satellite males that have higher testosterone are actually smaller. There's something funny about size and testosterone in satellites. I'm sorry, I'm gonna have to remember. I don't remember it clearly. That said, the circulating levels of hormones they're talking about are per volume, so not total blood volume. So they're certainly experiencing different doses, but it's true that the 3 male types are also different in size. So those two things always co-vary. And there's not enough overlap. So nesting males are really a separate size category. So it makes the allometric scaling a little bit tricky.
Maybe one more question and then we'll let you get to lunch.
Okay,
Sorry,
go ahead.
I saw Tyrone raise his hand 300 times
Oh,
on
I
Zoom.
Okay.
So I will
I'm
raise
so
my hand
sorry.
as for Tyrone, who was on Zoom.
All right.
Did nobody else see that? It's just like,
I think it just flashes if you still.
I'm so sorry.
okay.
Can you hear me?
Yeah, we can just go ahead and ask.
First of all, great talk, Suzanne. I had a couple questions. One, I wondered about how closely related your species is to the sex-changing wrasse, and do you see similar complications with male development and different types in the sex-changing wrasse? And also a technical question, I was curious about the flutamide results, that it decreased testosterone when typically, at least in mammals, you see an increase in testosterone with flutamide. So those are my two questions. Thank you, Michelle.
Yeah, sorry about that. I'm not a very good multitasker, especially with tech. So the symphytis wrasses, there's one of those wrasses that in that genus that there's some evidence that they change sex. It's also a cleaner wrasse. And they, I think you'll probably think of the labrids, like the thalassemia labrids are sex changers, sequential sex changers. And I think the best estimates, recent estimates of when they diverged from the symphytis, the labrines, the group I study is like 20 to 60 million years ago. So, it's a significant amount of evolutionary time. But what we know across, we've also done in my group some comparative work of the evolution of sex change. And so there's a lot of plasticity in the mechanisms underlying sex determination in almost all of the labrids. So these guys don't have sex change, but they likely have sex determination mechanisms that are not, simple genes or a sex chromosome. But the particular species I study are definitely not sex changers. So we've never found any evidence of sex change in their gonads. There's some others in the group that people have argued a bit about whether or not there's evidence they're sex changers or not. That was the first question. The second question is, so we found a significant decrease in circulating 11-keto-testosterone with the flutamide injection. And my understanding is that actually whether or not testosterone goes up or down depends a lot on the time scale of the impox. So you sometimes find short-term increases in testosterone and long-term reductions in testosterone with the flutamide. So there's a time scale, right? Because it's, as you know, right, there's feedbacks between the circulating levels of hormone, what's going on in the brain, and then the body's production of their own hormones. And so the studies that I've looked at go both ways. Sometimes you get a decrease in testosterone, sometimes you get an increase, and it depends a lot on the time scale at which those changes happen.
Do you have any idea of the mechanism for how it would decrease? Because typically with things like flutamide and tamoxifen, the levels go through the roof, like non-physiologically high.
Yeah.
So,
no, I don't know the direct mechanisms other than what we would glean from other vertebrates. Yeah, nothing in the wrasses themselves. Yeah.
Oh, thank you. Curious.
Thank you.
Well, the number of questions you had, you got, and the number of hands that were still up tells you gave a great talk. And so thank you very much once again.